I'm Hayden, I work in ESR. The project that I work on, it's the AIM2 project, so Aotearoa Impacts and Mitigations of Microplastics. We're a New Zealand-wide project. There's many different organisations and CRIs working with us, and we're just kind of doing a baseline assessment of what's there in New Zealand, some of the impacts they might have, and then in the future we're hopefully going to look at how we can mitigate some of these impacts. My role is the senior technician, so I do a lot of work in the lab, and my main thing is actually just quantifying the microplastics in different environmental samples. So we mostly look at bivalves, so we're doing a lot with the green-lipped mussels, which is obviously an important food source in New Zealand. We've also done some dolphin stomachs, some sediment, seawater sampling. We've done it all. It's great to get out in the field and do some of the sampling as well. This here's quite a lot of what we do in the lab. So we'll get a sample like this here. This here's just some sand, so a sediment sample. And microplastics, they're tiny in comparison to sand. So you want to somehow isolate the microplastics from that sample because you're never going to dig through and find all the plastic in there. So what we do is we use something this here that's called a density separation. So we've got a dense solution of salt in here, and that there causes the sand to sink because the sand's more dense than the solution, whereas plastics have a relatively low density, so they float to the top. So you can see some wee plastics in here. So then that's a great way to separate the microplastics from a sample. We do a couple of other things that aren't here, but we'll digest the samples if they're mussels, so we'll digest all the meat away and then we're just left with the plastic and so forth. And then we filter it down onto a filter paper. So this here's a seawater sample from Auckland, and then this here's what we then take under the microscope and manually pick out all the little microplastics that we find, take photos, characterize it for the length and the polymer type, color and all that sort of thing. And then that's what we can use that's our quantification of the plastics to see what's out there. There's a lot more known now, but when I first started, the thing that surprised me most was actually that there's plastic everywhere. So basically every sample I've looked in, I've found plastic. It's quite easy to find. All different size ranges, like you can't see most of it, it's all under the microscope. But yeah, so that surprised me a lot. And then considering we've mass produced plastic for well over 50 years, the other thing that surprised me is how little is actually known about the plastic. We see plastic as this kind of inert thing that's great to use and doesn't break down that, but then it does break down to smaller particles. There's all these contaminants associated with it and added to it, and just surprised me how little's known or even to public knowledge of what's out there. At the moment, we are just doing a lot of this quantification stuff, so still finding out what levels of microplastics are in New Zealand. So where I'd like to go next is looking at the impacts of these. So there's a heap of stuff added to these plastics, as I was saying, and we know they're nasty chemicals by themselves, but they're there in the plastic. Do they come out? Do they affect the environment, which then leads to human health effects and all that?